In our previous video, we have discussed about what is CFTR, that is related to cystic fibrosis uh, problem, and in this video we will discuss about what are the uh, pathogenesis of CFTRs. That is, what are the possible treatments or future possible treatments of CFTR. So, the CFTR is a gene that is responsible for cystic fibrosis problem. So, this is an example that is shown here. That as we know that the genes are present in DNA in the form for exon introns, and finally this after the splicing of mRNA, these introns are removed, and then these exons are combined together to form pre-mRNA. After that they will form a mRNA and goes into the and move towards the cytosol of the cells, where it is translated by the ribosome. And the red region is shown here as the defective region. So this defective or dysfunctional CFTR will not be able to perform its normal activity. So what are the possible uh, treatment or future possible treatment? First is the use of antisense oligonucleotide. It is generally used to block the formation of protein or to prevent the protein to work in a particular way. So this antisense oligonucleotide uh, strategy can be used to block this inherent part, or to change this inherent or to change this defective part into a correct mRNA sequence. So, and this thing has been tested, that is known as QR010. So this thing has been tested to correct this defect and form a corrected mRNA that is that finally can be translated into a repaired CFTR protein that is able to perform the required function. Third one is to do a gene editing from with the popular with the with the discovery of this CRISPR technology. This thing can be used to correct this defective position of the CFTR gene, and then the correct mRNA form can be used for the translation. Last one is the gene replacement. In this, the defective CFTR gene can be uh, converted into a cDNA with the help of reverse transcriptase or some other uh, process, and this complementary DNA then be corrected either artificially or in vitro, or they can be corrected in vitro with the help of different technologies, and then this corrected pre-mRNA can be formed, that finally converted into mRNA, and then the correct CFTR protein can be produced. Another thing can be used is that this defective uh, CFTR gene will form defective pre-mRNA, and then it will form mRNA, and this mRNA can be modified chemically to form a correct mRNA that can be translated then back to repaired CFTR protein. And this repaired CFTR protein will function as normal as uh, the actual CFTR protein. So here the first figure or the top figure shows how the defect of CFTR looks like, and second figure shows what are the possible cases. That includes the cases that we have discussed in the previous slide. This is the normal functioning of CFTR. In this case, this mucus layer is hydrated, and what happens is that this ENaC, that is epithelial sodium channel, and this chlorine channel will be controlled by this CFTR protein. As the chlorine channel decreases, it will gives a function to the ENaC to adjust accordingly. However, in case of defective CFTR, this chlorine channel is not to its has not been able to perform its function, and uh, there is no direct connection or direct we can say there is no direct coordination between this chlorine channel and this ENaC, that is sodium epithelial sodium channel. So what happens in this defective CFTR is that uh, since the ENaC is active and this chlorine ion is not active, the with time after certain time or in this defective CFTR, this ENaC become hyperactivated, and because of this, what happens is that this mucus layer get dehydrated and it become more thickened. So, and because of this, the mucus will not be able to perform its function, as the mucus is more thickened and it is not hydrated. How this thing can be addressed is the first one is that this problem occur because there is a formation of bacterial layer that prevents the chlorine that prevents the normal functioning of this chlorine channel. Because of this, the balance between sodium and chlorine is not been able to maintain, and then this ENaC, it is epithelial sodium channel, will get hyper activated. So in order to control this bacterial infection, the antibacterial biofilm can be formed that can prevent the formation of this bacterial biofilm, as this bacterial biofilm the will prevent the response of host immune system, and also it is resistant to the several antibiotics. So this antibacterial biofilm needs to be treatment needs to be given in this case, so that this bacterial biofilm can be removed. Another thing is that in which the because of the genetic disorder, this chlorine channel is not been able to perform normally. In order to treat this, we need to inject the correct gene of CFTR, either through viral vector or non-viral vector, and it can be either for a short duration of time. It can be translated directly in the form of mRNA, uh, and if the host genome integration is required, then it needs to be transferred it with the help of viral vector. Uh, and in this case, all the strategies that we have discussed in previous slides can be used in this uh, scenario. As it is shown that this oligonucleotide targets CFTR, it will correct the defective gene of CFTR, and this mRNA encodes CFTR. This is the case of transient transaction where mRNA can be directly translated in the cytoplasm of the cells, and this is the case of cDNA encodes CFTR. This is the third case that we have discussed in the previous slide, in which case it will uh, form a correct DNA sequence. It will form a complementary DNA sequence of the defective mRNA, and then be corrected, and then form a and then be corrected and then form a correct pre-mRNA and then mRNA, which can be translated to form a correct CFTR. And in the third case, what happens is that the third case, this because of the hyperactivation of ENaC, that is, as I have told many times, epithelial sodium channel, there is the balance between sodium channel and chlorine channel is not been able to formed. So in this case, the silencing RNA, siRNA, or the silence RNA has been used to inhibit this hyperactivated sodium channel, so that the balance between sodium channel and chlorine channel can be established, and then the CFTR will perform normally. So these are the strategies that can be used, or in, that can be used in future to treat this CFTR problem, defective CFTR, to treat or cure this defective, treat or repair this defective CFTR gene, so that it will not, so that patient will not face this cystic fibrosis problem. That's all for this video. Thank you for your time.